Good afternoon, everyone. I'm Yunjin Go, a master student from Aja University School of Medicine in South Korea. I'm deeply honored for the opportunity to present my research at the ASCS 2024. Today, I'll be discussing the finding of our study titled HepScope CNN-based single-cell discrimination of malignant hepatocytes. Our investigation centers on hepatocellular carcinoma, the most prevalent form of liver cancer, ranking as the sixth most common and third most lethal cancer globally. Despite the advancement in treatment modalities, the prognosis for HCC remains bleak due to the intrinsic heterogeneity of tumors. This underscores the critical necessity of delineating the precise tumor microenvironment using the single-cell data. While novel techniques such as single-cell RNA sequencing and spatial transcriptomics have emerged, but there remains a lack of specific markers or gene sets capable of accurately identifying malignant hepatocytes within HCC. Even established markers exhibit a limitation in this regard, and recognized tools like InfraCMV can help discern malignant cells in cancer single-cell RNA-seq data. However, this tool is very time-consuming, and it predicts for DNA-level information from the RNA level. Hence, we aim to develop a novel gene set and an AI tool to address these challenges, hoping to contribute to the accurate identification of malignant hepatocytes, as well as the discovery of therapeutic targets. Firstly, using InfraCMV, we delineated the malignancy status of hepatocytes in a large integrated single-cell RNA-seq data consisting of more than 34,000 hepatocytes from normal and adjacent HCC and HCC liver tissues. And next, we performed the differential gene expression analysis using Wilcoxon rank-sum test between the malignant and non-malignant hepatocytes. With a predefined log-to-fold change and adjusted p-value, we identified 113 genes significantly upregulated in malignant hepatocytes, which make up our HepScope gene set. And sorry, it's not working. Yeah, I pressed it, but it's not. Oh, thank you. Yeah, now it's working. The gene set encompasses 77 novel genes compared to existing HCC-related gene sets, which are derived from bulk RNA-seq or microarray data. And we observed its capacity to distinguish malignant hepatocytes in both the internal and external validation single-cell data. Moreover, we validated the robustness of our HepScope gene set on the bulk RNA, proteomics, and spatial transcriptomics data as well. Similar to results from single-cell RNA-seq data, HepScope performed comparably or even better than existing HCC-related gene sets. Notably, our gene set successfully discriminated between tumor and non-tumor cells in spatial transcriptomics data. Furthermore, HepScope also emerged as a significant survival prognostic marker in both univariate and multivariate analysis, with samples obtaining higher risk scores showing a worse survival. Encouraged by this finding, we sought to leverage the distinctive expression patterns of each of the 113 genes within HepScope gene set for even more precise prediction of malignant hepatocytes. We happen to observe a structural similarity between ECG data and gene expression metrics. Hence, we opted to employ 1D convolutional neural networks commonly utilized for time series data such as ECG or automatic speech recognition. To mimic the structure of ECG data, we ordered the HepScope genes based on descending log-to-fold change values. Comprehensively, we trained and tested the model by using normalized expression gene expression matrices from 11 single-cell RNA-seq data. To ensure the sample number balance from each class, we performed the random sampling for malignant hepatocytes. So in total, the model was trained with 8,000 hepatocytes, and then validated and tested with 4,000 cells. And furthermore, to avoid the overfitting or vanishing gradient problem, we adjusted the hyperparameters, for example, learning rate or optimizer, and we applied the early stopping mechanism as well. We first demonstrated that the HepScope trained model outperformed the models trained by other HCC gene sets as supported by better AUC and APBJC score. Subsequently, we confirmed the superiority of our model structure over alternative models. We converted 2D convolutional layers to 1D convolution layers and standardized the hyperparameters across other models for fair evaluation. So HepScope CNN demonstrated superior performance and balance across the evaluation metrics, including time consumption, as compared to other models. Lastly, we sought to investigate how differential expression of HepScope genes may influence the tumor immune microenvironment in HCC. To do so, we stratified 121 samples based on ascending HepScope module scores of malignant hepatocytes. Samples in the top and bottom 25% were designated as HepScope high and low, respectively. Subsequently, we calculated the proportion of various immune cell types. Interestingly, in the HepScope high group, there was an increase in the proportion of CD40 cell and B and plasma cells, while the proportion of CD40 cells and NK cell, dendritic cell were decreased. Further annotation of immune cell subtype revealed that HepScope high tumor immune microenvironment harbored more immunosuppressive cell types compared to HepScope low TME. Particularly noteworthy was the approximately twofold increase in the proportion of regulatory T cells in the HepScope high niche. Conversely, the proportion of cytotoxic immune cells were more abundant in HepScope low TME. Additionally, our cell-to-cell interaction analysis unveiled specific signals within HepScope high niche, predominantly associated with extracellular metrics or tumor progression. Remarkably, we identified a signal that has not been reported in HCC, LTB, and LTBR, occurring between regulatory T cells and malignant hepatocytes. This signal is known to induce the migration of regulatory T cells in mouse liver, suggesting that HepScope may not only predict malignancy, but also shed light on the tumor immune microenvironment in HCC. In summation, the HepScope gene set featuring 113 genes represent a significant advancement in understanding the complexities of HCC. Its utility extends beyond identification of malignant hepatocytes, encompassing prognostication and comprehensive characterization of tumor immune microenvironment. Moreover, our study pioneered the use of convolutional neural network model for gene expression analysis in HCC. While our study presents groundbreaking findings, there remains room for further validation. Yeah, I extend my gratitude to my colleague Jasmine Ting Ting, who shared first authorship, and to Professor Darren Ting Lim and Professor Subin Lim for her invaluable guidance and support throughout this endeavor. And I'm also grateful to my landmates for their constructive and continuous support, and I welcome any inquiries or opportunities for collaboration via email. Yeah, thank you for your attention.